Hi, everyone. We hope you had a great break, and we're getting ready to get started into session 3, bioengineered tissues. And so today our moderators are Dr. Nuala Del Piccolo from UC Davis and Dr. Vitor Gaspar from the University of Aveiro.
Hi, I'll be introducing our first speaker, which is a PhD student from the University of Cambridge, Anthi Mosidou. So please correct me if I mispronounced that wrong. That was all right. Thank you. And the title of her talk is, sorry, I had it up on my screen a minute ago, is a 3D bioelectronic model of the human intestine. So I will hand it over to Anthi.
All right. Can you see my screen and hear me all right? Great, perfect. Thank you for the kind introduction. Hello, everyone. My name is Anthi. I'm delighted to share my PhD work with you this afternoon on a bioelectronics approach to build a 3D in vitro model of the human intestine. In our group, in Bioelectronic Systems Technology Group, we use organic electronics to build in vitro models with integrated electronic elements for label-free and dynamic monitoring and sensing. Our approach is based on iterative improvements of both the biological systems and the electronic devices in a parallel process. And we now have a toolbox of bioelectronic devices that can be predictive of real biological systems, such as cell membranes on chips or more complex 3D cultures and tissues. On the material side, we work with conducting polymers, which have been shown to be particularly apt for interfacing with biology. And mainly we work with P.PSS, the structure of which is shown here on the left. So P-dot is a P-type semiconductor with mobile holes on its backbone that can hop from one chain to the other. These holes are then compensated by the sulfonate anions of PSS, which can be considered as ionized acceptors. So with P-dot, a device we use a lot is the organic electrochemical transistor, and here is the typical structure of such a device. Basically, a thin film of P.PSS is in contact with an electrolyte in which the gate electrode is immersed. Then metal electrodes called source and drain establish contact with the film and define the channel through which flow from the source to the drain. This is the on state of the device, and such devices work in depletion mode. So upon application of voltage at the gate, cations from the electrolyte will be injected into the film, and this will lead to a drop in the number of holes extracted from the source to the drain, and eventually the current we measure at the drain will be also decreased and the device will reach the off state. It's like having a switch where the gate voltage is the input and controls the output. Now, when cells grow on the channel, they impede the ionic flux from the electrolyte to the conducting film, further modulating the electrical properties of the material. And what makes OECTs very attractive for our applications is that they are inherently powerful amplifiers of the transduced signals. So what we can do with devices such the OECT is, for example, to measure the activity of electrogenic or non-electrogenic cells, monitor critical parameters like cell coverage or the formation of barrier in real time and in a non-invasive manner. And the good thing with thin films of P.PSS is that they are optically transparent, so we can do combined live imaging. However, we're now moving from these planar devices and we're engineering more complex 3D biological systems fully integrated with adapted electronic elements. And my PhD work falls under this scope. Particularly, I'm working on the Gut-Microbiome module of our IMBIBE gut-brain axis in vitro platform. And today I'm going to show you the progress I have made on building the intestinal module of the platform and the electronic device that hosts it. So in terms of electronics, my model is based on a novel device our postdoc Dr. Pizzalidis recently developed. Essentially, he created a 3D OECT inside a tube, a transistor in a tube that we call Tubistor. The configuration and the operation principle of the Tubistor is similar to the conventional OECTs, but the difference here is that the channel is no longer a thin film of P.PSS, but we have a three-dimensional tubular electroactive scaffold that is capable of hosting and concurrently monitoring 3D cell cultures. Building on this work, when I started designing the module, I turned my attention to the structure and function of the native tissue and to see essentially how we can improve the physiological relevance of the system. So looking at the intestinal tract structure, as part of the gastrointestinal tract, we have a hollow tube with thick walls that consists of several layers of cells, each of which has a distinctive function, but essentially supporting the innermost mucosal layer where enterocytes and goblet cells and other cells form the intestinal barrier that separates the laminal contents and the commensal microbes from the rest of our body. And focusing on the functional barrier and the microenvironment, we decided that we have to modify our device a bit. So we came up with this new version of the device, incorporating the laminal compartment of the intestinal structure. As you can see here in the SEM and X-ray microcomputer, the tomography images, the channel now is a hollow, tubular, highly porous electroactive P.PSS scaffold. And the new device is called the L-Tubistor. So in my approach to reconstruct the desired tissue microenvironment in the L-Tubistor, I was based on gradually building a triculture cell model that consists of intestinal cells and fibroblasts, the former to form the epithelial barrier and the latter to form the lamina propria layer underlying the intestinal barrier in the native mucosa. So in the first seeding stage, this first seeding stage corresponds to cultivating fibroblasts in the porous bulk compartment of the scaffold to form this underlying connective tissue, but also to fill the pores and thus block the infiltration of the bulk compartment by intestinal cells injected a few days later and guide them to line this laminal surface of the scaffolds and therefore form the intestinal barrier as they would in vivo. So in this second stage, we inject KECO2 cells and HT29 cells, and now we have a triculture system that we maintain for another 3 weeks to allow for differentiation towards a polarized intestinal epithelium, while of course we continuously monitor this electrically. Moving on results now, but just to mention that I'm only sharing findings from static experiments today. Firstly, optical analysis showed that during the first stage of the culture, the fibroblasts successfully infiltrate the bulk of the scaffold after 4 days in culture, lining the walls of the pores, as well as secreting proteins such as collagen, laminin, and fibronectin, forming an in vivo-like extracellular matrix network, nicely setting the ground for the subsequent cultivation of intestinal cells and therefore fulfilling the role I gave them in this triculture system. At the end of the experiment, pretty much after 4 weeks in culture, immunofluorescence staining for typical intestinal markers, including zonulin-1 in green and mucin in red, effectively showed that our P.PSS scaffolds effectively supported the growth, maintenance, and differentiation of intestinal cells towards a uniform epithelial layer on the lumen lining of the L-Tubistor. In addition, Z-stack confocal images and H&E staining showed that this epithelial layer is polarized and it is formed actually on top of the fibroblasts of the lamina propria-like layer formed from fibroblasts, as we can see here in both the H&E staining and this snaps we have from the Z-stack confocal images. However, What makes the L-Tubistor to stand out as a model is the ability to monitor closely the establishment of both tissue types throughout the course of the experiment. And we can do this, we can monitor this by operating the devices both as electrodes and as transistors. And we were able to confirm through these the presence, adhesion, and growth of cells in the porous electrode. Here in this slide, we can see the evolution of the output characteristics of the L-Tubistor when we operate them as transistors. So here on top, we observe a decrease in the maximum saturation current over time, which can be attributed to the decrease in the conductance of P.PSS as a result of cell activity. What I mean is that as cells gradually form the tissue on the porous electrode, they partially block the ion flow from the electrolyte, which in this case is the cell culture media, to the 3D channel. They add resistive and capacitive elements on the scaffold, and thereby they significantly affect its electrical properties and the performance of the device. This is also confirmed by the decrease of the transconductance over time. This is another figure of merit we use with transistors that reflects the magnitude of the amplification of the transistor and hence its sensitivity to transduce the biological events we are trying to monitor here. These findings are further substantiated when we perform the devices, when we operate the devices as electrodes, so electrochemical impedance spectroscopy in this case. This showed us a significant increase in the overall magnitude of the complex resistance of the scaffolds during the formation. It shows the response here in more detail of the electrode, of the porous electrode, to the modulation that cells perform in this case. We see the shift in the overall impedance as the tissue is formed, and then the impedance reaches a plateau at the last stages of the culture. The phase plots here also confirm the addition of capacitive and resistive elements. We can see here considerable decrease of the phase maximum at low frequencies and the presence of a typical time constant in the mid-frequency range, which are characteristic for these types of cells. Essentially, what I want to point out is that this bimodal inline monitoring, which is of course cross-validated with optical analysis, provides us with more comprehensive electrical readouts, which can reveal valuable information for the biological model, but in real time, non-invasively and without requiring labels. So my take-home message with the example of the L-Tubistor as a different approach to build organs on chips is that bioelectronic systems bring unparalleled capabilities in building and dynamically monitoring tissue engineering models. I didn't show any data today, but we're currently working on integrating fluidics to operate the devices under dynamic conditions, which will also facilitate the integration of the microbiome module of our IMBIBE platform. And of course, we want to substitute commercially available cell lines with stem cells or even organoids. And with that, I would like to thank my lab and collaborators. Thank you all for your attention and the organizers for putting together such a wonderful symposium. Happy to answer any questions.
All right, it looks like we have a few questions here in the chat. Thank you for a beautiful talk. Hector is asking about your choice of cell line. Said if he heard correctly, you used HT29 cells.
Yeah, because it's sort of a proof of concept, we're trying to build in parallel the device and the biology. We decided to go with commercially available cell lines KECO2 and HT29 for the intestinal cells, which are the cell lines that are used for many years now when people are building in vitro models of the intestinal barrier. And the fibroblasts we have are telomerase immortalized fibroblasts, if this answers the question.
He is asking about why you chose that cell line instead of a non-cancer cell line. And his second question is whether...
Both cell lines in literature have been shown to be spontaneously differentiating into healthy intestinal cells. It's what
Oh,
is
perfect.
deadly. So.
Mm-hmm. And the second question is whether you tested if the HT29 cell line expressed mucus.
Yeah, they do. In the pictures, if I may go back to
Certainly.
that, let me see. So yes, here the red is mucin. So red on top of the polarized epithelium is mucin.
Oh, perfect. Thank you. I think that answers the question. And then we have a couple of questions about the electrical field. Can you talk a little bit more about what pattern of electrical currents you applied and what effect they had on the cells?
Electrical currents, I'm not sure I understand that. And what we essentially do, because these polymers are conducting polymers, they're capable. There is current, even if we don't apply any voltage, any current, anything on the material, there is current there. So the devices at the beginning are on the on state, there is current there. What we do is we apply a voltage to push cations from the electrolyte, from the cell medium, into the tunnel. And what this does, it means that holes from the material, this current that the material has at the beginning is decreased. So the output, when we measure the current in the output, we see this decrease in the current. So the device goes from the on state to the off state. It's
I
the
see.
accumulation mode. There are other materials, N-type materials, that work in accumulation mode, where then you operate them slightly different, in slightly different manner to have an effect. Does this answer the question?
Yes, thank you for clarifying that. Vitor, were you keeping time? I'm not sure how much time we have left for questions.
Yes, in the interest of time, I think we should move on, Nuala. It was a very nice presentation by Crescenti. Let's move on to the next speaker. Thank you, Crescenti. Very nice.
Thank you.
Our second talk will be presented by Dr. Zhanbo Zhang, who is at MIT, and he will be telling us, the title of his talk is Organoids-Derived Gut Microphysiological Platform for Host Microbe Immune Interactions. And I'll let you take it away.
Thank you for the introduction. Can you hear me clearly?
Yes.
Perfect. All right. First, I would like to thank the organizer for pulling out such a great program. And I have learned a lot along the way to listen to the talks. So today I'm going to share the device that our lab has developed to study the crosstalk between the gut colon mucosal barrier and microbiota. And as you have heard, as you have heard in many news headlines that gut microbiota has been positively associated with many diseases, and these diseases either occurred locally in the gastrointestinal tract or remotely in other organs in our human body, such as liver, brain, or lung. However, many of these studies are carried out in animal models, such as non-abiotic mice. And as you would probably agree that despite many important similarities, mice is very different from human in terms of like anatomy, genetics, and physiology. So it cannot fully recapitulate a human system. And this is particularly true in the terms of gut microbiota. And here you can see that this is a distribution of different bacterial species in mice fecal microbiota and human fecal microbiota, shown in red. And as you probably have noticed that many bacterial species are very specific to human hosts, where you cannot observe any of them in animals. And more importantly, many researchers have tried to have attempted to inoculate this bacterial species into the animal models has failed. So there is a pressing need for an alternative model in order to allow us to study the interactions between this bacterial and human microbiota and a human body. So, so back in 2015-16, two talent postdocs, one from Professor David Champer's lab, Jun Yuan, and the other one from Linda Griffith's lab, UJA, together with great technician John Cammett, took this challenge and start to invent a physiomimetic platform, we call it, Gut-Microbiome-Immune physiomimetic platform, in short, GuMI. And this is a picture of the prototype at early 2017. So after many generations of iteration and troubleshooting, this device has evolved to a piece of art, in my opinion, where here you can see on the top of the figure, where everything is designed to accommodate standardized transfer where many labs have used it regularly. And you have the baselateral unit to recirculate in the media to support the growth of mammalian cells. And in a apical side, you have the apical unit and to supply the bacterial media, which is anaerobic, to support the bacterial growth. One important feature that I would like to highlight in this device is that the height between the colon epithelial cells to the top of the apical compartment is about like 3 millimeters. And this is significantly higher than other devices, microfluidic devices that you have seen. So because of this, we call it, instead of microfluidic, we call it mesofluidic device. And there's a big advantage for this because this will significantly decrease the shear stress during the operation of the flow. Therefore, it allows the many bacteria can reside in on top of the epithelial cells, which I will show you later. So to establish the whole primary human colon model, what we do is to isolate the colon tissue and maintain the colon stem cells as organoids, as you can see it here in matrix gel. And then afterwards, we dissociate the organoids and then see them as a monolayer and then introduce into the device. And this is done with a huge help from two great technicians, Charles and Kirsten, and in collaboration with Ilma's lab at MIT and David Brown's lab at Boston Children's Hospital. So I have got a lot of questions that people often ask, why you want to use the monolayer instead of a 3D structure and organoids and so on. Well, my argument here is that when you look at the autonomy of small intestine and large intestine, where in the small intestine, you have a very big villi and relatively small crypt where the stem cells reside. The villi in the small intestine is created to like expand the surface area to for the nutrient uptake among other functions. But in the large intestine or colon, it is mostly flat with a very deep crypt. So the flat surface here is the interface of the bacteria and the colon epithelial cells. So it is arguably that monolayer would be more appropriate for the colon. So moving forward, so we introduce these monolayers into the device and culture them in the oxygen gradient for 3 days. And as you can see here, after 3 days in the device, the colon epithelial monolayer seems to be very intact still. and they are very clear cell borders. And if you do the immunostaining with a MUC2, you can clearly see there are many MUC2 positive cells, presumably goblet cells, as seen in vivo. And also, very importantly, these colonocytes are differentiated with a correct polarity, where you can see a preferential expression of NHE3, which is a mono carboxylic acid transporter that are observed in vivo in human colon. So after to this point, we were very excited about the device and what we could achieve. And then the next to which material we want to introduce. And to do that, we went to the literature and asked, what are the most abundant species in human gut microbiota? And here you can see the top 30 most abundant species in a human fecal microbiota in terms of relative abundance and the prevalence. And among all these bacterial species, one bacterial that is particularly eye-catching to us is called Faecalibacterium prausnitzii. And this is because there are many studies have shown that this bacterial is the depletion of this bacterial is positively associated with inflammatory bowel diseases such as ulcerative colitis. And more importantly, there have been studies showing that this bacteria is particularly sensitive to exposure to air or to oxygen, surviving for less than 2 minutes. So for us, this is an ideal candidate to demonstrate the capability of our device to create or maintain the oxygen gradient. So, so we did that, and this is how it looks like once you introduce the Faecalibacterium prausnitzii with the colon epithelium. So clearly you see a very big cloud of bacterial cells on top of the epithelial monolayer. And very interestingly, you can see there's a clear gap between the bacterial cells and the epithelial cells. And presumably, this is a very tight mucus layer where the bacteria cannot penetrate into the mucus layer to get contact with the epithelial cells. So after demonstrating this, we were very excited and keep going to ask, what are the bacteria are doing? What the bacterial cells are doing? So we did the RNA sequencing for the mammalian cells, for the epithelial cells, in 2 conditions. So in the presence of the bacteria versus no bacteria, and to see what this bacteria is modulating. It turns out there are surprisingly more than like 4,000 genes that was significantly changed solely by this bacteria. And keep in mind that there are only 20 to 25,000 genes in human genome. So this is a huge amount. And among all this, I don't have time to go through all the data, but one very interesting pathways that we observed is the NF-kappa B inflammation pathway. So here you can see that with the bacteria, we were able to see that the activator of the NF-kappa B inflammation is downregulated by the bacteria. And in the opposite side, the inhibitor of this NF-kappa B inflammation pathway is being upregulated. Therefore, this is real data. Therefore, it provides let's say, a higher resolution how the NF-kappa B pathway is being regulated by this bacteria. And this is just a very one example of how the bacteria can modulate the human biology in the model. And I have to highlight that many of this observation is in a great agreement with the observation in clinics, which is very encouraging. And I envision this model could be useful in a broader aspect. For example, for anyone interested in disease modeling of gastrointestinal tract, such as a celiac disease, bacterial infection, this model could be perfectly useful. And also for the female reproductive health down on the bottom, such as the bacterial vaginosis, this model could also be useful to modeling those. And also, the device could accommodate the bacterial therapeutics or prebiotics to test how this influence or interact with the bacterial microbiome and the human physiology. With that, I would like to thank a lot of people, especially our collaborators. This is a huge collaborative project. So the Professor David Champer, who's leading the mechanical engineering effort, Chris White and Oma Ilma's lab, as well as Professor Rebecca Carrier and Professor David Brock. And of course, we are very grateful for the funding from the NIH and the company Boehringer Ingelheim. And if you, so for the sake of time, I only show you a little bit of the biological data. If you want to know more, we have the paper is just online last week, so you can have a look on that. And if you want to listen more topics, what Linda Griffith's lab is doing, there's tissue talks tomorrow, so you can go and register to hear more about what our lab is doing. With that, I would take any questions.
Thank you, Jean-Bo, for a really, really interesting talk. I think we've all heard a lot about the microbiome in the gut in the last few years. So this is a really neat model.
Yeah. Thanks.
Let's see. Marco has a question. How long can you culture the system after you add the bacteria? Quite curious about that.
Yeah, so for the data I've shown you, it is about like 2 days in co-culture and 3 days in total. But we have tested so far 4 days co-culture, like 5 days in total in a system. But I speculate that you can go longer. It was just like very labor intensive if you want to maintain all of them. So yeah.
Sure. And you have to take this, I'm just curious, you said the bacteria is very sensitive to oxygen. So you just have to take them directly from a cryovial?
How
does that work?
We first culture them in the anaerobic chamber where the microbiologist is often used in a daily basis to culture the isolates from human gut microbiota. And then we place the anaerobic chamber very close to the device. So when we want to inoculate them, we just like do it very quickly to inject the bacteria into the device. Yeah.
Great. That makes sense. Vitor, did you have any questions?
I think we have lots of questions in the question and answers panel. I think Jimbo can answer some of those in the interest of exchanging science. It was a great talk. So much. I think we will move on. Thank you so much.
Thank you.
Thank you, Noala. So hello, everyone. My name is Vitor. I'm a postdoc from the University of Aveiro in Portugal, Compass Research Group, and I will be moderating the next 2 talks of this very interesting question, following up on Noala's moderation. Thank you so much, Noala. So we'll be starting off with Ishita Jain. I think I pronounced it correctly, Ishita. She will be presenting her work. She's from the University of Illinois at Urbana-Champaign, and she will be talking to us about microenvironmental arrays and computational modeling to delineate signaling and biomechanical cues in the pattern differentiation of liver progenitor cells. Ishita, looking forward for your talk. You can start.
Can everyone see my screen and hear me?
Yeah, it looks great. Yeah, looks great. Thank you.
Sorry for the long title, but hi everyone. Thank you, Naveed and Pam, for giving me this opportunity to talk about my research. It's been a lovely day, and I look forward to all the other talks as well. So today I'll be talking about microenvironmental arrays and computational modeling to delineate cooperative effects of notch signaling and biomechanical cues in liver differentiation. So the liver is made up of multiple hexagonal shaped lobules with the central vein in the center and hepatocyte quads radiating outwards towards the portal tride. The portal tride is composed of the portal vein, the bile duct, and hepatic artery. Hepatocyte cords are single cell sheets of hepatocytes that secrete bile salts into the bile canaliculi, which is then collected by the bile duct. During differentiation, liver bipotential progenitor cells, the green cells in the image, form the ductal plate around the portal vein. During the course of development, they further differentiate into hepatocytes, or the brown cells here, and cholangiocytes, which form the bile ducts, or the dark green cells shown here. Understanding the mechanism of this differentiation can help us better understand congenital diseases such as Alagille syndrome, in which patients do not have bile ducts, as can be shown and seen in this histology image, where the healthy patient has bile duct here, but no bile ducts here. Further, it has been shown that mutation in Notch ligand jacket 1 and the Notch 2 receptor has been shown to occur in this syndrome. Also, similar mechanisms are also involved in repair and regeneration processes in chronic liver diseases. For example, here is an image of ductular reactions, which is irregular bile duct proliferation, as shown by the brown CK19 marker, which is a cholangiocyte marker. Our lab has been extensively studying the influence of various biomechanical and biophysical cues in understanding this differentiation process. To do this, we use embryonic mouse bipotential liver progenitor stem cells. And when we culture them at 100% confluency under differentiation condition, they can differentiate into hepatocyte, as shown by the albumin stain here, or cholangiocyte when treated with TGF-beta1, as shown by the OPN or the cholangiocyte marker. Further, we showed that this TGF-beta induced cholangiocyte differentiation is not only dependent on just the soluble treatment, but the stiffness and the matrix composition of the substrate. For example, here, when cells are cultured, the person-positive cells for OPN on fibronectin is much lesser than the cells cultured on collagen 4. Further, when these cells are treated with blebistatin, which is a myosin 2 inhibitor, the differentiation decreases on almost all the matrix compositions. Lastly, we also show that this TGF-beta signaling goes hand in hand with Notch signaling, as shown here by increase in expression of DLL, increased expression in the ligand DLL1 and JAGAD1. In the process of understanding this complex mechanism, we also discovered pattern differentiation of these progenitor cells when they were cultured on a well-defined circular geometry. Here we see that the cells on the periphery expressed majorly OPN, which is a cholangiocyte marker, whereas the cells not on the periphery expressed HNF4 alpha, which is a hepatocyte marker. Further, we can do a quantification as shown here, where we show radial expression of both these stains in a line graph. So you can see that HNF, which is this red line, decreases right where at the periphery, one being maximum radius, so the periphery, and OPN increases right where HNF starts decreasing. To understand this model better, I decided to computationally model it. I wanted to make a Notch-mediated model, since Notch has been shown to be implicated in many patterning events during development. And further, when we ourselves treated these cells with gamma secretase inhibitor, which is a Notch signaling inhibitor, we showed an increase in OPN levels, and hence showing a possible role of Notch in this differentiation pattern. So to do this, first, I simulated cells on a circular geometry. In addition to that, I incorporated the cell substrate tension and cell tension. And lastly, in the model, we do consider these two different ligands with distinct functions, DLL1 and jagged1. I started with an established model in the literature called the Collier model, which is just simulation of Notch signaling of cells in a regular lattice. The simulation starts with equal concentration of Notch ligand and the receptor. During the course of the simulation, we end up with a checkered pattern where the blue cells represent high Notch ligand and the white cells represent high Notch receptor. I modified this model by adding transactivation and cis inhibition. Transactivation is when ligand on one cell interacts with the Notch receptor on another cell, leading to the cleavage of the intracellular domain, or commonly known as the NICD, and downstream Notch signaling. Further, cis inhibition is when ligand on one cell inhibits the receptor on the same cell from interacting with the ligand on another cell. Hence, in that cell, there's no cleavage of the NICD and no further Notch signaling. As I mentioned before, I wanted to incorporate the cell-substrate interaction. So to do that, we have devised in our platform to measure the traction force data experimentally that is being incorporated into the model. And the cell-cell interaction is being modeled as an increasing function of the number of neighbors of a cell. To expand a little on this, on the periphery, there are high traction forces and lesser cadherin forces. Hence, transactivation was modeled to be a function of traction forces, where the Notch receptor on the peripheral cells could interact with jagged1, leading to NICD cleavage, hence more Notch signaling, hence cholangiocytic fate. The cells in the center experience more cadherin forces, less traction forces, hence cis inhibition was modeled to be a function of cadherin forces, where the Notch receptor was inhibited possibly by DLL1 to interact with jagged1, hence lesser Notch signaling, hence no cholangiocyte fate. Here I have the simulated modeled cleaved NICD levels of in cells just on this circular pattern. I have not yet included traction and cell-cell forces. We see a similar checkered pattern where the green cells represent high NICD and the blue cells represent low NICD. When I just incorporate the cell-cell forces, a similar checkered pattern appears, but the overall signaling is lower because probably just cis inhibition is occurring. Further, when I include traction forces, we get a thicker pattern of active NICD, kind of just restricted to the periphery. However, when I include both the traction and the cell-cell forces, it is definitely restricted to the periphery and correlates well with our experimental observations. One of the good things with this model is that we can perform many perturbations to this feasibly, and they can reveal useful insights. So the first thing I did with this model was to create Notch ligand in silico knockouts. When I decrease the DLL1 expression in the model by 90%, I see an increase in the active NICD on the periphery, whereas the opposite happens when I do the same with the jagged1 ligand on the in the model. I see no active NICD at all. Interestingly, when we did the same things in our experiments, where we knock out these ligands in our bipotential progenitor cells using lentiviral transfection of the SH RNA, we see similar results. When we knock out DLL1 knockout, we see a stronger pattern, as can be seen in this line graph as well. And when we knock out jagged1, we see no pattern at all. Another interesting thing was that this not only affected the cholangiocytic differentiation, but it affected hepatocytic differentiation as well. When we knocked out DLL1, the hepatocytic differentiation just dropped down to 0, whereas it reduced a bit in jagged1 knockout. In order to understand the cell interaction and the cell-cell cell-substrate interaction well, we stained for EC-adherin, actin, and YAP. EC-adherin is a cell membrane protein that forms dimers at cell junctions. It was shown that EC-adherin was found to be higher on the periphery. However, it was mostly intracellular, as can be seen in this zoomed in image. Actin, on the other hand, was mostly common across the island and fell right at the edge. YAP, which is a protein that localizes to the nucleus upon mechanical stress, was also found to be higher on the edge. Overall, all these interactions gave us interesting insights as to how these two factor cell-cell interactions and cell-substrate interactions might be working towards the differentiation treatments. Another way to perturb this pattern differentiation system was soluble factor treatments. For example, here, when these biopotential progenitor cells were treated with EGF, we see an increase in differentiation in the wild type in general. Further, when the same was done in DLL1 knocked out cells, the increase in differentiation happened everywhere, as can be seen with this increase in OPN positive cells in the center. Further, we also treated these cells with TGF beta 2 and saw that the increase in differentiation was actually more in the wild type than in the DLL1 knockout cells. So the two different treatments revealed different insights, which is interesting. In short, summarizing my work, we were successfully able to generate patterned Notch signaling model, which with incorporated cell-derived forces. Transactivation was modeled as a function of traction force, which was more on the periphery. Hence, Notch was able to interact with jagged one, leading to more NICD cleavage and Notch signaling on the periphery. On the center, cis inhibition was modeled as a function of cell-cell forces, cadherin-based forces, which was probably leading to inhibition of the Notch receptor to interact with jagged one, and hence no NICD cleavage. So all in all, experiments and this model showed us insights about how these two ligands might be working in this differentiation process. Further, the EC-adherin, YAP, and actin showed interesting patterns, and perturbation to the pattern were also interesting with these two treatments. Further work along these lines would be to incorporate the soluble factor treatments into the model, and possibly find a mechanistic connection between Notch and hepatocytic differentiation. And lastly, we look forward to translating similar model to other cell patterning systems. In the end, I would like to acknowledge my PI, Dr. Gregory Underhill, our funding sources, and thank you all for listening to my talk, and I welcome any questions. Thank you. Oh, sorry. Yeah.
Thank you so much, Ishita. Very, very nice talk, bridging the gap between in vitro and in silico modeling. Very interesting. So I guess we have a couple of questions in the Q&A answers panel. The first question is from Pablo, and he was wondering whether you are modeling the effect of mouse cells. If human cells would behave something more like a human person, what is your opinion on this? Why did you use mouse cells?
Oh, so we look forward to translating the system into using human cells. We haven't yet had the opportunity of acquiring possibly iPSC cells. It's very hard to get your hands on human stem cells, like primary human stem cells, because the concentration is too low, and that's why it works really well with this cell line, which is kind of a rare cell line, like having an embryonic bipotential progenitor cells. You can get hepatocytes easily, human hepatocytes or mouse hepatocytes, but yeah, we do look forward to getting some human iPSCs and then maybe differentiating them and using them.
Yeah, that would be very nice to see in the future. And the next question is from Stephanie Campbell. Actually, there are two questions from Stephanie. I guess I'll go for the first one. And she's asking, how does the organoid model work, and which component of the SEM did you find it works the best? And what media have you used for your work? What type of culture media did you use?
So we use RPMI media, which is, I think, common RPMI 1640 with normal FPS conditions. I'm not sure about the SEM question. We do not do any SEM measurements. And the organoid system is a contact printing of matrix on a polyacrylamide hydrogel, and then the cells are cultured on it. Yeah, I think I covered that. So it's a 2D based system.
Like 2D based system. Okay, okay. So there's a couple of more questions in the Q&A answers, Ishita. I think you could answer them just in the concept of exchanging scientific knowledge. And I think we will move on to the last speaker of this very interesting session. Thank you so much, Ishita. And it's my pleasure also to present Dr. Abhinav Sharma. Abhinav is coming from the University of Massachusetts in Amherst. He will be speaking about dynamically responsive gut mucosal barriers on a microfluidic chip. Abhinav, I will pass it on to you.
Thank you so much, Vitor. Hello, everyone. So I'll be presenting my PhD work on developing a microfluidic chip. I hope you guys can hear me properly.
Yeah, it's perfect, looking good.
Perfect. So it looks like wearing a mask is a political statement. So I'm gonna give my talk while I'm wearing a mask and see if that works for everyone. All right. So just to introduce myself, this basically shows my entire journey. I was born up here in the foothills of Himalayas, did my bachelor's down here in the middle part of India. I went to the northeast of India to get my master's degree, so a full triangle. My parents are actually logged in right here. There's no road still connected, but they have internet, so that's great. So today I'm going to be talking about gut, and it's great that the stage has been set with some great talks in the session before, so I can even just skip my intro slides, but I'm going to just discuss a little bit about how complex this environment of the gut is, where we have exogenous materials that come from our diet, bacteria that survive in your food. Then we have some endogenous secretions such as gastric acid, bile acid, and we know that gut is filled with close to 100 trillion bacteria per gram of the fecal content. And I think most of you know that 70% of our immune system is located in this area to take care of any sort of infection. So overall, the gut has all of these functions where it helps us maintain homeostasis with the external environment. It enables immunity development throughout our lifetime. Most of the nutrients and drugs taken orally are absorbed in the gut, and most of the metabolism occurs here. So if this balance breaks down, a lot of things can happen. So dysbiosis, which is basically just disproportionate microbiota, if you have more pathogenic bacteria compared to your commensal bacteria, which can lead to chronic inflammation, diseases like IBD, or molecular malabsorption that leads to malnutrition in a lot of countries, and a whole bunch of other diseases. So because of that complexity, it is very important to develop in vitro models of this system. And to speak broadly, I think we have already seen two models. So one is a very static type systems where you have transvaal systems. And we saw the talk before where they have converted this also into a flow-based device, but most of the researchers do static cultures where this membrane supports epithelial cells. We can co-culture this with immune cells. And we have dynamic systems such as microfluidic devices and gut on a chip type systems. So if you look at it broadly, I think most of the static systems are used for conducting drug absorption and toxicity based type testing, whereas the flow systems are good for host microbiome interactions because you can circulate the bacterial medium so they don't overgrow. So the most important factor that prevents us from this whole lot of complexity, all of these pathogens and acids, is this mucus layer that essentially coats the entire surface of the epithelium down here. And this paper showed this very nicely, where in a wild type mouse, this red bacteria were nicely separated from the epithelium, which are in blue. So this is a healthy condition. And when you knock out the gene that is responsible for mucus secretion, MUC2 in the intestine, you see bacterial penetration throughout. So what is this mucus made out of? It's made out of this awesome protein called glycoproteins called mucins. And they have this very rich chemistry. A lot of interactions can happen because of these carboxyl ends. So a lot of electrostatic interactions. You can have hydrophobic interactions. You can have hydrogen bonding within the mucus, so it cross-links. So you have to prepare the drug carrier or any type of drug that you design. You have to take into account all of these properties so that it can be absorbed pretty well. Now, the problem is that human intestinal mucus is not available. So I'm still waiting for the day when humans start to donate intestines and researchers can access them in a very short period of time. But in the meantime, a lot of research has been done where it's been shown that porcine mucus or pig mucus is very similar to human mucus. And we have done studies, these are all published. So the first study, what we did was we established a very simple method to extract porcine small intestinal mucus. So if I say PSIM, this is what I mean. And then we incorporated that mucus with an epithelium layer in a transvil platform, co-cultured that with immune cells, and we were able to recapitulate the colonic mucus layer in vitro, where if you see the in vivo mucus layer in colon is close to 850 microns. And with PSIM at 20 milligrams per ml under this conditions, we were able to get the same thickness of the mucus. And here you can see we conducted studies with salmonella and a mixture of probiotic to study competitive growth inhibition. And we found that salmonella was able to escape this growth inhibition because of the mucus. Mucus prevents most of the commensal bacteria or probiotics, but salmonella has developed mechanisms to go through this mucus, and we were able to show that in this image on the right. So this talk is mostly about developing a microfluidic chip. So imagine that static system. If you can flip it onto its side, most of the systems are developed in a vertical sense. So you have to change the focus to look at interactions at different points. So my idea was to develop a system that was all in a single plane. So it's a planar format, have an extracellular matrix down here, which will support the epithelium growth. And then we can flow in either mucus, and we can flow in luminal components, can be drugs, bacteria, or anything. So these are the goals that we were looking to achieve with this. So to incorporate all of major types of the intestinal cells, develop a fabrication technique that is simple and high throughput and could eliminate cost and time in fabrication, automate this so we can image different interactions at this interface in real time and be able to establish flow of the luminal content. So to do this, we developed this very simple technique where here you can see a plotter cutter just cutting away, so we can cut a whole bunch of these devices. At the same time, I can go have my coffee while this prints my devices. Then we put it into a plasma cleaner, so it can be bound to a glass slide. And once it's bound to a glass slide, so here you can see it turning pink, this is plasma. And once it's bound to a glass slide, then we are ready to develop the system further. The trick here is to save the PDMS pieces that we cut out of the in that step. The reason is we can put those back after plasma bonding, and then we introduce a collagen matrix. This is type 1 collagen from rat tail. And here in this video, you saw me introducing that manually, and then take off the cover that was preventing the collagen from flowing in after it has formed the gel. So it forms a very stable gel down here, and we can run multiple devices at the same time. So here you can see about 4 times 4, 16 times 2, 32 devices at the same time, and we can put close to 8 devices on under the microscope, and we can do parallel experimentation with this. So for the next step, we wanted to see if the collagen gel would enable diffusion of the molecule so that if we have cells embedded inside the collagen gel, they stay alive. And we found that within 4 hours, we get to the steady state of diffusion of molecules of the size of 20 kilodalton, even as high as 155 kilodalton, that's TRIC down here. And we found that embedded immune cells remain viable for more than 7 days. Now, this allows us to conduct these co-culture studies where we can introduce bacteria. We can track the growth over time, so you can see the intensity of bacteria growing over time. And what we found is that without bacteria, most of these cells remain within the gel, but with introduction of bacteria, these cells were coming out of the matrix and they were starting to attack the bacteria that were present there. So the next step was now to create the interface of that epithelium. So what we do is we introduce the cells, single cell suspension into the device. We keep the device vertical so that all of the cells fall onto the interface of the collagen and then allow it to culture for 1 or 2 days. Once they're attached, we can culture it in either parallel configuration or vertical configuration. And within 7 days, you can see that we see this 3-dimensional growth. These are HT29 cells, and they naturally grow in 3 dimensions. So there's no flow. We are culturing these under gravity-driven flow. The next step was to introduce the mucus. So I showed you before that we have this process to extract PSIM, the pig intestinal, small intestinal mucus. And now we can look at different conditions. So mucus is known to interact with pH. So in our stomach, that's how our stomach is protected from the acid that's secreted. So it forms this thick gel once a pH 2 solution is flown in and the mucus is flown from the bottom. Mucus also interacts with cations because it has a net negative charge. So we know that with calcium, it would aggregate. So you can see this interface that forms, and it's dependent on how much calcium I put in this channel. So you can see the thickness of that interface change. So now you can see that if I flow in bacteria without any calcium, they just flow in and then over time start to diffuse all the way down to the bottom end. Whereas with calcium, you can see the bacteria getting trapped up here, and in the entire device, there's a clear separation between the bottom part where there's little or no bacteria, the top is full of bacteria. So you can create this nice separation after I introduce this epithelium. So the bacteria is on top here, Salmonella, that is tagged with GFP. This is the mucus layer that separates this. And if I stop the flow or this barrier breaks down, you can see the bacteria coming in direct contact. So this shows that we can get that. So this device now can be used to then study these real-time interactions. So here in the video, you saw bacteria interacting with epithelium and then start to break the barrier and diffuse into the tissue, and that can be all quantified. We also looked at no bacterial condition. You could see that most of these are whole blood immune cells. And you could see that none of these cells are moving too much, whereas in presence of bacteria that's tagged in red, E. coli. So we saw a lot of these cells come out of the matrix and try to compete with the bacteria. So all of these things can be easily quantified because you can visualize them in real time. Here you can see a cell trying to go out. It looks like a neutrophil. So if we have techniques where we can tag different types of cells with different colors, we can easily track what cell is responding at what time. So we get a nice spatial and temporal information of these cellular dynamics. Then you can also see how bacteria is flowing under gravity-driven flow without mucus. But in presence of mucus, you would see the direction of the bacteria is starting to change. So they're kind of in the matrix now, and they're moving downwards towards the epithelium. Now, the epitome of this entire model is if we can add in circulating cells, so we can also incorporate adaptive immune system along with our innate immune system that would be present within the tissue. So here, this device shows that we are able to culture epithelial on one edge, endothelial on the other edge. These are HUVEC cells, and HT29 again for epithelium. We can introduce the immune cells down here, And this movie now shows a nice rolling and attaching phenomenon with the endothelium. So now you can imagine adding these adaptive type immune cells with stimulation on the top, if they can infiltrate into the tissue, and we can quantify all of those responses in real time with this device. So in conclusion, I think we have achieved most of the goals that we set before we started. So we are able to capture the dynamically responsive mucus layer that has the thickness matching the physiological thickness of the colonic mucus layer. And we can change that by just changing the mucus concentration that we have. And that was enabled by optimized extraction of the functional mucus. We are developing chips with very simple, high throughput and cost-effective manner so that anybody in the entire world can easily replicate these studies or use these for specific hypotheses. It enables real-time imaging and quantification of these cellular interactions. And as I said, these are universally adaptable. The next step could be personalized models where here, as an example, I've shown you an organoid taken from a mouse intestine that is infected with salmonella, and you can see overnight how it kind of explodes. These can also be co-cultured, so we have seen organoid type models in our previous talks. But if you disperse these cells now, and they can be taken from any different organ, and after we take out those cells, we can either embed them inside the collagen or any type of matrix. So we also looked at talks that were talking about different types of matrices. Or also the vascularization would be really interesting within this, with 2 channels, and any of the organs can be mimicked by just a single platform. And I hope that this could be utilized in future for, say, COVID-19, testing drugs and vaccines for COVID-19 and looking at mucosal responses from those vaccines. With that, I would like to thank you very much for this opportunity. I think this is a great platform for all of the trainees, and thank you to all the people that are present.
Thank you very much, Abhinav. It was a very interesting talk, very interesting technology. And we have a lot of questions in the Q&A panel. I will try to get to some of those. In the interest of time, I think we have time for 2 questions. And the first question is from Carla. And it asks us if you could use this device to analyze neutrophil transepithelial migration.
Absolutely. So you already saw that we were able to establish the epithelial layer. I know that this is a kind of a multi-layer, so if we can create a monolayer of epithelium and have embedded cells that are able to do those extravasations, absolutely we can capture that phenomenon easily here.
Very nice. Thank you so much, Abhinav. And the other question among one of those is, are the bacteria affecting the structure of the collagen at all? This was a question that I also have. Do the bacteria affect the collagen?
That's a great question. So I tested 2 different strains. One was E. coli and Nissl, which is a probiotic, did not diffuse into the collagen as much, whereas Salmonella was able to go through pretty quickly, and they were able to swim really fast. So I think it would be interesting to see. It's easy to open this device up, so we can do some mechanical characterization, we can do SEM, other type of characterization to actually look in deeper into that. So that would be a great thing to do in future. So this is so simple to do. I think anybody should be able to test it.
Should give it a try, right?
So
okay, thank you so much, Abhinav. We will only ask you if you could answer all the questions, okay, in the interest of sharing science, okay?
Yeah, absolutely.
And thank you very much for your presentation. And I think this wraps up our very interesting session, Nuala. This is a very pleasure to have shared this session with you. I believe we have an interval of 5 minutes, and I hope to see you in the next session. Thank you so much.